And welcome to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we're really happy you're here today. And we will be discussing design principle in the beyond rule of five space with Henrik today. Henrik Moebitz from Novartis. Welcome, Henrik.
Thank you, Dennis. Glad to be here.
Yeah. Henrik trained as an organic chemist and completed his PhD in biochemistry at the University of Freiburg in 2003. And he was fascinated by enzyme catalysis, but he still remains convinced that enzymes are the better chemists. So Henrik started his postdoctoral training at the UC Santa Barbara in California, focusing on molecular mechanics of enzymes. And in 2005, he joined the Department for Computer-Aided Drug Discovery, abbreviated as CAD, at Novartis in Basel. And Henrik is a very passionate drug hunter who knows how to integrate physics-based property predictions, structure-based drug design, and data analysis into any drug discovery project. He's also able to bridge the interface between chemistry, biology, and pharmaceutical development, and has developed deep expertise in targeting helicases, kinases, and specifically the MAP kinase pathway. Currently, he co-leads the CAD group in Basel and oversees multiple medicinal chemistry projects. Henrik is actually the author of more than 50 publications and patents, with a significant body of his research still to be published. So Henrik, that's a very, very impressive CV. And yeah, I was blown away by your talk. Actually, we briefly talked about it in the discussion at the ACS in New Orleans. I think that's one of your greatest achievements, and I think one of my favorite drug discovery programs ever, actually. The HRO Werner Helicase project.
Thank you very much. You're embarrassing me, and I don't know, I didn't put so much goodies into my CV, but you embarrassed me a little by picking out the goodies. Thanks a lot for your kindness.
Sorry about that. Yeah, we usually start with some icebreaker questions. So I just wanted to ask a few questions, like what inspired you to start a career in drug discovery?
Well, I've had actually a lifelong fascination and passion for chemistry. It started pretty early on when I was a kid or a teenager. My first brief fascination for high energy chemicals, lead azide and stuff like that. And I quickly had a lab in the basement, and I did things like crystallizing copper sulfate and running to pharmacies. At the time, you could still buy benzene and dangerous chemicals over the counter. That lab got shut down after some time because of smell occasions, and I actually moved on to already to the university. And yeah, I mean, retrosynthesis and Corey's red book saved me from boredom in high school. And so I've been fascinated by chemistry all my life, and just following my interest and fascination naturally led me to biochemistry, and because I like challenges, it became clear that understanding or connecting structure to function was a key step, and this is why I landed in modeling, because the structural biologist wouldn't want to have me as a biochemist. And ultimately, in drug discovery in the industry, I joined Novartis in 2005. And really, this is a job where all of those passions come together, and my interest in chemistry, structure, complexes, and now also physical chemical properties and all of that. I mean, it's a dream job to be combining all of that into medicinal chemistry and the biology, of course.
Awesome. Awesome. Yeah, no, I can very much relate to that. I also had this kind of chemistry kit, the C3000, if you remember that. So
Yeah,
I tried the phosphorescence and fluorescence concepts in our bathtub also.
yeah.
But yeah, my mom was like not having that.
No.
And yeah, I can also relate to the great group at Novartis. I've been working there for about 7 years and the Emeryville side. So yeah,
Mhm.
you have a really, really great team, and especially the computer-aided drug discovery team that you're co-leading in Basel has made a huge impact in Emeryville, and yeah, on I think, in my opinion, on drug discovery as a whole.
Absolutely. Yeah, we're an incredible team of cadders, not just
Awesome,
in Basel.
awesome, awesome. Yeah, so I mean, the next question would be, what is your favorite molecule and why? I kind of know
That's
the
obvious.
answer, but I still
I
ask.
I actually have one that I like even a little bit better, but
Oh, okay.
that's a story for another day. But HRO is a bit of a miracle if you consider all of the things we had to do to get to that point. So the fact that we were able to find something after a lot of struggle and with the properties that it has in that space is, yeah, it's quite a bit of a miracle still.
Yeah, no, you're absolutely right. This is a very fascinating. The way you developed this, the way you deeply thought about those concepts and implemented that into practice is utterly fascinating to me. I'll tell you that.
Thank you
Yeah.
very
So,
much.
what are you most excited about in drug discovery today?
Um, when I joined, we couldn't compute much molecular weight. There was structure-based design, there was virtual screening, similarity works, but I think what we've been able to increasingly calculate more and more, and that makes for fascinating improvements in our ability to drug things that were undruggable previously. And I think we're getting dealt more and more challenging targets. So necessity is the mother of invention. We have to be better than just sort of generic structures, and we have to kind of sprinkle pixie dust over our molecules, and we can now do that not just with a lot of technologies. I mean, I mentioned the advancement in computational chemistry, but there's of course also a lot of advancements in our biological tools and high throughput biology, and just all over our tools are getting sharper.
Yeah, yeah, definitely. Even AI we're seeing now, but I think yeah, that's something that we have already used for a while in I guess in drug discovery. What's your opinion on AI really quick off the
That's a loaded question. Let's just say that for the talk that I'll be giving, there's no neural networks were harmed.
Okay.
So it's more
Okay.
physics based.
We'll leave it there. Physics based is yeah, we like first principles, right?
Yes, yes.
There you go. All right. So if you could go back to kind of the beginning of your career and give yourself some career advice, what would you tell your younger self?
Um, don't be afraid, follow your passion, and I would probably do it over the same way. When I started, I had a fascinating mentor, a very dedicated medicinal chemist who told me a lot of also properties, and he was also leading from the bench, Harry Rüger. And I looked up to him, and I would never imagine to be able to tell a story such as HRO. But yeah, it's basically just following interest and passion, and not getting bogged down by the organizational and scientific challenges.
Yeah, no, that can sometimes happen in large organizations, and yeah, scientific challenges everywhere. But yeah, as you said, our tools are getting sharper, molecules are getting more complex, right? So now we're able to target more challenging targets, things that we haven't been able to target before. All right, yeah, before we launch into your talk and I show a brief summary of HRO, last question, what do you like most about the Drug Hunter?
You make it really easy to follow a large body of literature. I do like to think of myself as an avid reader, but usually a big chunk of the molecules of the month I missed. So, and they went often into the study. I really like finding things I missed and having things put into context that I didn't find or have the interest to follow up with reviews. For instance, the GLP-1R summary, that was a very nice perspective.
Awesome. Yeah, thank you so much. We really, really appreciate it. All right, yeah, before we launch into Henrik's presentation, let's have a look at Henrik's favorite molecule here and the case study that we actually published on Monday. And Henrik graciously reviewed the case study for us. So yeah, here you can see this complex beast of the Werner helicase, which is an allosteric inhibitor. We give you an introduction here as to why you should be reading about this, and all these purple links that you see here link out to primary literature, but also it goes beyond that. It goes into patents, company presentations, websites, clinical trial pages, and all of that. We give you a little bit of a perspective as of why it's so difficult to target helicases, and Henrik can probably sing a song about this. We're showing you here what is the competitive landscape, what are other Werner helicase inhibitors, how was this particular one discovered, how did it lead to the hit initially, what was done here in the lead optimization phase, what were the challenges that were faced and really nicely solved, and then some really beautiful concepts that Henrik's team has worked out here with non-classical Zwitter ions, as well as the introduction of the neutral TPSA that we'll be hearing more of today. There's a preclinical profile of the compound. We dive into the binding mode and how that binding mode differentiates from the binding mode of other, maybe covalent Werner helicase inhibitors. Give a bit of the background of the preclinical data on Werner, on specifically animal models, and also a little bit, since this is early days, a little bit early clinical development, as well as the synthesis here, which is also very complex for a molecule like HRO 761. And with that, without further ado, Henrik, we're really looking forward to your talk about design principles in the beyond rule of 5 space. Everybody is excited about this.
Okay, well, I mean, thanks for the introduction, also for the nice summary of the Werner story and HRO 761. This project actually has been a trip, and I can speak not only for myself, but also for my fellow team members. A big shout out to the chemistry team here, Ross, Jürgen, Joseph, and Vincent, being the key actors. I think we've really unlearned a lot of classical chemistry and learned a lot of new concepts. And some of those concepts were so general that I felt that they deserved being explained in standalone publications, exemplified by marketed drugs. And as you're all aware, the rule of 5 doesn't predict oral bioavailability. It rather predicts the lack thereof based on insufficient permeability and solubility. And in this talk, I'll be following the same subtractive logic, but using 3D PSA and logP as proxies for those two parameters. Those of you who work in the beyond rule of 5 space also know that the rule of 5 clearly doesn't work anymore. And so the question is, what other rules are there? What thresholds can we use? And what are the design principles? And not only is it important to predict each of these properties, so permeability and logP, but more importantly, how do you combine high permeability and low lipophilicity? This is still the sort of stated goal of the rule of 5, because there are two properties that are anti-correlated. We've also just published a second principle that Dennis alluded to, so non-classical Switter ions. And this is a fascinating phenomenon that is exemplified in many drugs and with publicly available data, but it's been hiding in plain day sight, and it was hidden by dogma in a way. And dogma is also an aspect of today's talk. This brings me to the chameleon, which you can see here. Because chameleonicity is a concept that is often evoked to explain the oral bioavailability of beyond rule of 5 drugs. And the thought is that chameleonic molecules can adopt different conformation in water versus apolar environments, for instance, by forming intermolecular hydrogen bonds. And that this leads to an overestimation of the polarity by TPSA relative to 3D PSA. And the difference between these two parameters, TPSA and 3D PSA, was suggested as a quantitative descriptor of chameleonicity. Interestingly, the Switter ion dogma and the term molecular chameleon, they were both introduced in the literature by the same person, Bernard Testa, who is one of the godfathers of modern PhysChem characterization. However, I mean, chameleonicity is a great concept, but most of the chameleons that you find in the literature are actually red herrings. I'll explain later why they are basically computational artifacts that are due to bad physics. And I'd like to thank, if you can press again, the editors of Chem for allowing me to put that message on the front cover. On the next slide, I want to illustrate again why I'm talking about balancing lipophilicity and permeability. And I want to illustrate this with the challenge that we were faced with in the Werner program that led to HRO 761. So for an oral drug, you need three things: permeability and solubility for absorption, permeability to get into the cell, because most targets are cellular, and potency on the target to get an effect. This plot here, which I call the LO map, combines all of these three parameters. So you have permeability on the x-axis and biochemical lipE on the y-axis. And this is a snapshot from an early stage of our optimization when we were prior to LO, molecular weight already mid 550s. And we're looking here at matched pairs that are differing in a substituent that faces towards solvent. So this is a region of the molecule where you can introduce polarity without negatively affecting potency. So you can see that we can change polarity by varying the substituent and thereby reducing logP and increasing lipE while maintaining more or less the potency. But this comes at the cost of potency, and 1 log of permeability corresponds roughly to 3 logs of biochemical lipE. And as you can see illustrated by the cross-diagonal lines, you can also clearly map where the clinical candidate needs to be, because you have your TPP from the start. You know, that's going to have to be highly permeable and highly potent with a reasonable lipE. And the question was, how can we ever get to the top right if all we do by balancing polarity and permeability is moving across the cross-diagonal line? Actually, as we will see in a few slides, most of the compounds, I don't know, can I get the laser pointer to work? Can you share the laser pointer?
Yeah, let me see. Maybe I can give you remote control. Did you get control now?
Yes. Okay. Can you see my pointer?
Anyway. I
Then I'll
can.
just
Sorry.
I'll just have to voice over. As you will see, most of the clinical candidates in the beyond rule of 5 space are actually in the lower right quadrant. So they are permeable, lower right, permeable, but very lipophilic. And on the next slide, I just have a few definitions. So beyond rule of 5 is just, in a way, molecular weight space between 500 and 1200 free Daltons, that's cyclosporine. So we're excluding prodrugs and permanently charged molecules, antioxidants, dyes, what have you. The bulk of this talk is based on a ab initio conformational analysis, and more on that on the next slide. And then the Cosmo RS, logP, and 3D PSA are compared between experimental values and structures, and we then extend that comparison to all compounds in the Novartis archive with measured permeability and logP. Three short definitions. So the permeating conformer, which also has the minimum 3D PSA within 2 kcal per mole in decadiene, that's assumed to be the permeating conformer. The minimum energy conformer in water is just what it says. This is the most populated water. And as proxies for both, we use the PDB because that's a binding in a polar environment, or the CCDC, where the molecule sees its own mostly greasy environment. And last but not least, I'll show you that this measure of chameleonicity, which I introduced, is actually better viewed as something else, which I call neutral polarity. And on the next slide, let's briefly compare, and maybe you have to click again. Okay, let's briefly compare the Cosmo RS generated 3D PSA and also logP with conventional 3D PSA. So the RESCOS workflow is a workflow that my colleagues Anniko Udvarheli and Rainer Wilken, together with Stefan Rode, developed. And this combines all number of standard conformer generation tools with a clever clustering and selection algorithm that also uses scoring in different solvent environments, and then leads to a set of relevant conformations that are optimized by QM methods, the most expensive that we can afford, so on a DFT level. And these conformations are then used for physichem property calculations with the state-of-the-art tool Cosmotherm. And so that gives you logP, pKa, Boltzmann distributions, which also allow the calculation of strain, etc. And in most cases where we've used this, you get a concordance of measured and predicted properties. You also get some outliers, and that is often an invitation to better understand the chemistry of the compound. The non-classical Swidler-ions is an example, or maybe to realize that you actually made a different compound, or that you have an artifact in the measurement. Conventional 3D PSA, on the other hand, is basically just counting atoms across conformations that are force field generated, and therefore not accurately energy scored. So you might have very high energy conformation, and that might give a high spread of 3D PSA, which is not physically meaningful. Here I've just, for illustration, used the same conformation, the minimum energy conformation of my favorite molecule, HRO, that we already talked about. And you see the Cosmo RS sigma surface on the left versus the conventional 3D PSA, where all the polar nitrogen, oxygen, and hydrogen atoms are shown on the surface. And that is almost twice as high in the 3D PSA that you get out. And TPSA, in the end, it's nothing else than TPSA fit to conventional 3D PSA. On the next slide is a look at the physical properties of marketed beyond rule of 5 drugs, oral. We have about 113 in our archive of the 154. And of those, only 95 could be measured in logP. And we've even used more expensive assays like shake flask or potentiometric logP. And some of those compounds are so poorly behaved that you cannot determine a logP because of a lot of experimental issues due to the poor physical properties of the compounds. You can see that the correlation of Cosmo-RS predicted and direct logP is pretty decent, R-square of 0.6. Of course, this data set is heavily biased to the upper right of, or the sensitivity limit of the assay, which is 4.7. For some individual compounds, we do measure higher in individual assays, but for the bulk, 4.7 is the detection limit. And you can see that the majority of the compounds is above that range. The good correlations also allows us to extrapolate that to the entire set. And here you can see that about 50% of the drugs are logP greater than 5. That's on the top right pie chart. And only a few of them have only about a third of them have a logP in the optimal range of 3 to 5. Moreover, many of them are ionized. So you see about 80% of them are acidic, basic, or Swidder ionic. And the number of neutral species increases in those that can be measured. So many compounds just achieve a log-D of less than 5 by ionization. On the next slide, we see that this is likely due because this data set has a bias for permeability. The compounds have to be permeable because most targets are intracellular. And here's the trend between minimum 3D PSA and decadiene and measured MDCK-LE. I have to mention this is a version of the assay that is engineered to cope with beyond rule of 5 compounds. So we include BSA in the receiver compartment, and that increases or decreases the problem of retention in cells, something that is an artifact in many permeability assays. So you may have seen a correlation or a bell-shaped correlation of permeability with logP. And this drop-off of permeability at high logP is an artifact due to cell accumulation. In this assay, this is not present. And you can see that most of the compounds are in the high permeability bin. And actually, we also here observe the same thresholds that were previously postulated for rule of 5 compounds. So the red line is 140, that's the Weber limit. And the gray line is 100. This is a value that we found predicts quite often more than 10. And this is also was also the value that was found in a Pfizer publication. The trend, although weakly correlated, reflects that was seen in the landmark paper from 1997 on the right, where PSA is sigmoidally fitted to human bioavailability. There are a lot of reasons for the poor correlation. We have diverse chemical space, ionization states, and very different lipophilicities here. But you can see that the geometric mean between those compounds below 100 and above 150 is strikingly different. And if not anything else, then this 3D PSA allows you to rule out ideas that have a high chance of being impermeable. What is also implicit in this plot is that there is a high disconnect between TPSA and 3D PSA. because some of the compounds here have TPSA up to 250 or 300. And the highest values that we see here are slightly above 150. And so because we have the accompanying conformations, we can look at the difference between the 3D PSA in different environments, which is on the next slide. So here you have the minimum water 3D PSA and the minimum 3D PSA in decadiene. And this is color-coded by the difference in intramolecular hydrogen bonds, with green being the maximum up to 3. And you can see that in most cases, there is no change of intramolecular hydrogen bonds. The exceptions is for the class of HCV NS5A inhibitors, which have a proline urea, proline imidazole motif that actually does have environment-dependent intramolecular hydrogen bond. But this is relatively rare across the whole data set. And you can also see that there are few disconnects between the two 3D PSA measures. All data cluster very closely around the unity line. The plot on the top right is another version of showing that. And even the ones where I mentioned that have changes in intramolecular hydrogen bond, the change is only roughly 10 square angstroms per intramolecular hydrogen bond formed in decadiene. And if you look at the totality of the difference, this only makes up a small fraction of the neutral TPSA. So in a way, TPSA that's not accounted for by the 3D PSA. You can see that on the plot on the lower right. But of course, you might object, these are just computed conformations. We don't have solution conformations. So what about experimental conformations? And this is the same comparison on the next slide between PDB conformations and CCDC conformations as proxies for polar and apolar environments. And you see the same thing. So it's a near unity correlation. Here we're looking at the medians of 50 beyond rule of 5 drugs that were present in both databases, again, color-coded by the differences in intramolecular hydrogen bonds. You can see here that the difference is most compounds don't have a big difference. And again, the few exceptions that have high differences on the top right graph only have something like 10 square angstrom per intramolecular hydrogen bond. In fact, here you could say that cyclosporine is the exception, which has been generalized to the rule of chameleonicity, but the entirety of the data set does not possess appreciable chameleonicity. On the next slide, let's look at how these trends hold across the larger Novartis data set. So this is about 10,000 compounds with measured permeability. And although this is a sort of a cheaper version that was of QM that was used to generate these 10,000 calculations of the minimum 3D PSA decadiene, we still see sort of a similar trend recapitulated. You may argue whether bifunctional degraders marked in red have a slightly different trend and maybe also higher thresholds for high permeability. But you can also see this highly populated cluster with high permeability just below the 100 threshold. And on the right is how a plot of the increase of 3D PSA with molecular weight. You can see it's basically a linear function of molecular weight with a certain fluctuation around that, around the linear regression in bold line. And interestingly, if you were to plot the 3D, the TPSA expected for this, it crosses the 100 PSA, 100 square angstrom PSA threshold just as the molecular weight of 500. And so you might think whether the 500 Dalton in the rule of 5 is actually just an encoded version of this 3D PSA threshold. The reason why I can plot that in is shown on the right, because across is shown on the next slide, because here you have the distribution of the fraction of polarity. So that's TPSA divided by molecular weight. It's a measure of the polar atoms in a molecule, or the fraction of polar atoms. And across different data sets, this is remarkably close, around 0.2. And of course, this is biased by the chemical matter that is inside that data set. And if you drill down to beyond rule of 5 drugs and permeable beyond rule of 5 compounds in our data set, on the next slide, we see that they have similar thresholds. So on the right, you have the Novartis beyond rule of 5 compounds that are highly permeable, and they're populating a range of 0.2 to 0.3 square angstrom per Dalton. And this is the same that is also found for the beyond rule of 5 drugs. Interestingly, the compounds that are marked here in red, they are so-called oral beyond rule of 5 drugs, but many of them are actually gut restricted. They're just called oral because they're given orally, but they're actually intended to just stay in the gut. And those tend to be very poorly permeable. And you can view them as decoys in a data set that is otherwise very much biased towards high permeability. Now, the polar fraction must be correlated to logP. So let's look at that on the next slide. So there is almost sort of a bimodal distribution between 0.1 and 0.3. And the upper half of that, so this is shown here on the left, so the TPSA divided by molecular weight greater than 0.2, this has a median logP of 2.7 and only 13% with a logP above 4.7. So this roughly corresponds to the rule of 5 90 percentile, lower than logP 5. And you can see that the lower leg of the compound, so those with less than 0.2, is almost a log unit more lipophilic, and the portion of highly lipophilic compound increases by 2. And this trend is also found for beyond rule of 5 compounds. So it seems to indicate that this very simple TPSA by molecular weight greater than 0.2 enriches for low lipophilicity. On the next slide, you can see this combined with the minimum 3D PSA as a rule for permeability into what I call the rule of 1/5. This is actually, I won't bore you with the details because this is a bit the molecular equivalent of stamp collecting, but I'd just like to point out that the rule of 5, actually increases the fraction of high logP compounds relative to the non-compliant set. So this is an intriguing consequence of not considering that too low lipophilicity can also be a bad thing for permeability. On the next slide, we see a more interesting version of that plot. So this plot here shows, in essence, the green lines here demarcate the amount of neutral TPSA that can be generated by a polar fraction between 0.2 and 0.3. And this amount of neutral TPSA is required to maintain a 3D PSA of below 100, while also being predicted to have a logP of less than 5. And you can see that this green sweet spot here is populated by the majority of oral beyond rule of 5 drugs, 106 of 154. And by consequence, the compounds in the red spaces are either too polar or too lipophilic. And in these regions, we find compounds that have many iodines and are also known to be substrates, transporter substrates, or either really highly lipophilic. The number here is the logP. So all over, the oral beyond rule of 5 drug set that exists manifests a bias for permeability. And this is because this is non-negotiable for cellular activity. And we have to assume that the solubility is being fixed by formulation or ionization. But this is not really desirable. Ideally, you would have low permeability, high permeability, and low lipophilicity. On the next slide, we take a look at neutral TPSA and how it changes across chemical space. You can see that it sort of increases with molecular weight, but it's also highly biased by the kind of chemical space. For instance, the bifunctional degraders shown in red here tend to have a lower fraction of neutral TPSA, so that likely accounts for this dip. But overall, the spread in the data is almost as high as the dynamic range. So there's a wide fluctuation. And we can pick out some compounds that have a very high or low neutral TPSA. And for instance, the millerite is a compound with a lot of intermolecular hydrogen bonds, but none of them are dependent or change as a function of the environment. You can actually view this as a pseudo cycle. The compound 2, a kinase-like hinge motif, also has a high neutral TPSA. In this case, there are no intermolecular hydrogen bonds. And we also have 2 examples here of beyond rule of 5 drugs with high neutral TPSA, despite having no potential or no intermolecular hydrogen bonds here. So this is one suggestion that neutral TPSA is actually an intrinsic property of the molecule that is mainly a function of the structure of the molecule. And we can see an example of this in the next slide. This is from a PRMT5 paper. I know that's another target that you're excited about, Dennis. So here we have a series of PRMT5 inhibitors that we analyzed by a number of different analytical methods. So you have RESCOS computation on the left in white, then the NMR solution conformation in magenta, the small molecule X-ray in green, and the co-crystal structure, the binding mode, in blue. And you can see that across all of these bearing slight variations in the torsion of the aryl rings, we have maintained the same conformation of the core. So this is an example where we can really exclude conformational effects. And strikingly, if you click again, we not only see a modulation or a good correlation or trend between, in this case, hydrogen bond acceptor moment and permeability, but strikingly, we can increase the lipophilicity of these compounds by introducing polar atoms. So one nitrogen change from 1 to 4 increases logD, increases permeability, reduces the hydrogen bond acceptor moment, and introducing an additional oxygen atom further increases lipophilicity, permeability, and reduces the hydrogen bond acceptor moment. And all of these changes correlate with an increase in neutral TPSA. So this is clearly an example where an electronic modulation of acceptor strength is the root cause for this. And it also highlights the fact that we need to better differentiate polarity as an input and logP as an output of the molecule. And this, in a way, is what TPSA minus 3D PSA does. It adds in both the expectation, so the amount of polarity and polar atoms you put in, and the resulting polarity of the whole molecule into one number. On the next slide, we take a look at 3 first-in-class beyond rule of 5 drugs. On the top, you see the hits. on the lower hand, you see the first-in-class drugs or the first-in-class clinical candidates. Chiluprevir didn't make it to the market, but analogs of it did. And between these two, between hit and clinical candidates, there are usually only a handful of analogs which you can retrieve from the discovery articles. And I've plotted the neutral TPSA and the 3D PSA on the next slide for all of these campaigns. So in the left slide, in the left graph, you have the LO progress, so the progression of front runners leading from the hit to the clinical candidates, plotted against the neutral TPSA. And you can see that in all three cases, neutral TPSA increases. On the right, this is plotted in the LO plot. And you can see that all three campaigns in a way have the sweet spot as an attractor. They managed to get there regardless of where the hits came from. So for instance, daclatasvir was started from a phenotypic screening hit, which was a dimer side product. And this, being a phenotypic screening hit, this naturally had to have permeability. So this is why in this case, you don't have a large increase of neutral TPSA. But you do have that in venetoclax, which started from a merging of 2 FBS hits. And you can see here that the two first clinical candidates, 737, which had to be given IV, and navitoclax straddled sort of the 2 lipophilic border region and ultimately didn't make it in the clinic due to a lack of exposure and efficacy. On the next slide, we plot the HRO 761 LO campaign on top of this, which Dennis already glossed over in the HRO article. You can see here that neutral TPSA steadily increases, and we stay within the sweet spot during the course of the optimization. And because this was purposefully built in at the start, in the end, we also get the best balance of properties. This is shown here on the right. So HRO is really in the rule of 5 space with measured properties. As I mentioned in the beginning, I'm showing here calculated properties and not measured, because we do not have measured data for many of these compounds. So this is a better scale on which to compare things. On the next slide, this leads me already to the conclusion. So there are no new rules in beyond rule of 5 space. It's the same physics. So logP and permeability are really size independent, or the limits thereof. But the odds of achieving high permeability and low lipophilicity drop dramatically with increasing molecular weight. And this means that you have two options. You can either live with high lipophilicity and call for formulation to save the day, or you need to design in good properties carefully by, for instance, using minimum a neutral TPSA. The Cosmo RS 3D PSA of relevant conformations and the simple criterion of fraction polarity greater than 0.2 are predictors for increasing odds. And this can be very useful for screening large chemical spaces, which you need to screen to identify outliers that beat these trends of anticorrelated permeability and lipophilicity. The ruling concept of chameleonicity is not in play in most of the existing beyond rule of 5 drugs, but it's a really great concept whose potential still needs to be evaluated more with designed compounds. And lastly, neutral polarity. So the difference between a TPSA and 3D PSA is an intrinsic and size-independent molecular property and can be a very useful optimization parameter in beyond rule of 5 space, as I just showed. And lastly, I think we need a better language that differentiates polarity and also lipophilicity as input and output, because if we use the wrong language, this may prevent us from putting polar atoms where they might actually be tolerated because they're not that polar, as you can see, as you saw in the PRMT5 case. And on the next slide, I'd like to thank, first of all, the project teams that a lot of this work was done in Werner PRMT5 and also ErgDev. I'd like to thank Rainer and Aniko for the gift of Rescos and helping me troubleshoot it. Stefan, Damian, Tanguy, and Norbert for the measurement of that was shown in this talk. Jimmy for MolPipe that allowed Rescos-like parameters or predictors for our internal database. And lastly, Bernhard, Ruben, and Peter for very helpful discussions and input. And with that, really like thank you for your attention, and I'm happy to ask any questions you might have.
Awesome. Yeah. Thank you so much, Henrik. That was amazing. Like, I love that counterintuitive example, of course, of the PRMT5 inhibitor. So, yeah, I mean, in general,
the same goes also for methyl groups. So you can use methyl groups to reduce hydrophobicity.
So is there any way to like kind of, how would you predict this? Would you predict, would you start predicting neutral TPSA of these molecules? Yeah.
Yes. So, I mean, you can, I mean, the couple of things you can do. Empirically, you can do a matched pair lipE analysis. That will show you which compounds have a better balance of permeability and lipE. And if you plot them in a matched pair space, you see those outliers. They would break the trend. That is empirical. Computationally, you can either look for, again, in matched pair space, delta log P, delta 3D PSA. And neutral TPSA just happens to combine both things into one parameter, because TPSA plays into log P, as we've seen, and neutral TPSA plays into permeability. It, in a way, makes that polarity affordable while retaining permeability. And I can say that, yeah, in a way, in the end, in design, we're really looking for transformations, and most transformations don't do that. So you have to look at large chemical spaces, and very often the kind of transformations that do this are not even in the commercial space. So the hydroxypyrimidine acid in HRO was not even known in the literature.
Yeah, you guys did a great job in designing that, despite these cool implications. We got tons of questions from the audience, Henrik. I don't know if we're gonna have time, but for all of them. But let's start going through them. So I guess for the permeability in MDCK, is there a key modified conditions that work well in beyond rule of 5 molecules? Like, you know, people are suggesting extended incubation or the addition of 5% BSA. And is there a good beyond rule of 5 molecule that can be used as a reliable control for these modified assays?
Yeah, so this assay is described by my colleagues Felix Huth and from the group of Bernhard Faller. It's also cited in the paper. And as you've seen, we've been able to measure almost all of the compounds we had. BSA is one of the key components, but also checking for membrane integrity by adding a control. Because that's, I mean, a lot of those problematic compounds can also be soaps and destroy membranes. So you can have both false negatives and false positives. But this modified permeability assay was in a way triggered by beyond rule of 5 compounds, and my colleagues did a fantastic job of improving that.
Awesome. And do you expect to see beyond rule of 5 CNS penetrant clinical candidates soon?
Why not? I mean, in chemistry, everything is possible at a price. You can, HRO is an example that you can have a highly permeable molecule, and Wow, you can imagine that you could also dial down the efflux ratio.
Yeah.
But of course, the odds are smaller. That's clear.
Yeah, I think there are a few beyond rule of 5 CNS penetrant molecules or CNS active molecules, I think in the BTK space. And a lot of the times chameleonicity is invoked there. I guess it's a discussion for another day, Henrik. What do you think about this?
Yes, I've made my statement.
I think you did.
But I think chameleons are great. They're just not on the market yet.
Yeah, I guess we'll see hopefully them soon, right?
Yeah.
So another trying to use my Mac is the username of this person. I'm loving the 3D PSA permeability discussion, which certainly resonates with my experience. What about metabolic stability in the beyond rule of 5 space and in vivo clearance mechanisms?
There, I actually also looked into that. There's this concept of medlib-E. In a way, you have to correct for logP. You cannot compare compounds with different permeabilities. There is a signal there, but it's also not very strong. But we all know that reducing lipophilicity is key for clearance. And unbound exposure. And if people don't do that, for instance, in the bifunctional degrader, it's probably because of the bias for permeability. And neutral TPSA allows you to have that both low lipophilicity and high permeability.
Yeah, makes total sense. Were non-conventional intramolecular hydrogen bonds also included in the analysis of the 3D PSA? So that could be, for example, chlorine or fluorine shielding an NH bond?
I've used the definition from the Stahl paper, so oxygens and nitrogens. And I discuss also, or you can see that in the HRO 761 write-up. This is not just shielding, it's actually very complicated because yes, you have shielding, but you also have often acidification of the NH, and you have modulation of the acceptor that often comes after the NH. Could be an amide, could be an aryl. So it's a very really complicated, even in small modifications. So I think just saying this is an intramolecular hydrogen bond is not capturing the whole complexity of the phenomenon.
Yeah, makes sense. Can you comment on strategically how you position physics-based calculations such as Cosmo 3D PSA in active programs for compound design? When would you want to use physics-based calculations as opposed to, for example, a fast machine learning model for permeability prediction?
We use both. Of course, yeah, when you have larger molecules, a turnaround can be a week. That's still sort of compatible with normal assay turnaround times. So we do tend to involve physics-based methods more and more routinely. And I mean, the great thing about them is they also work for new chemistry.
Nice. Yeah, then I think Michelle is wondering what throughput do you have in this analysis and at what stage in the discovery pipeline can it be used to prioritize compounds? I guess that really depends on what kind of type of calculation you're using, right?
Yeah, so I mean, first of all, you can also pre-filter by using ML or TPSA and then kind of funneling it down. So actually, we've looked at about almost 1 million unique R groups in this program. So I once, for fun, calculated the chemical space that we looked at in silico, and the number is 10 to the 34 This is why I always smile when I hear ultra large docking spaces. So you can actually, by funneling it down and applying some tricks, look at very, very large spaces. In our database for HRO, we had something like 4,000 ideas evaluated. What's really just important is that you are disciplined and you're looking at matched pairs and not at random combinations, because that, of course, bites into the calculation time big time.
Yeah, it makes total sense. Then you have to recalculate the whole molecule rather than just this matched molecular pair change that you made. Yeah.
One more comment. I mean, obviously, sampling is a big challenge, and as not necessarily molecular weight, but number of rotatable bond increases, the challenge of sampling increases dramatically. So above 800 Dalton, it starts to get tricky, and you may have to work with tricks like truncation and alchemical cycles, etc. So, for instance, cyclosporin is an example of a molecule that I've not been able to calculate the experimental conformation.
Yeah, I guess that's the only chameleon out there, right?
Well, HRO is another one. I mean, but well, I mean, Baker's lab has shown that they can do that with that they can actually predict peptide macrocycle conformations quite well.
Makes sense. And we're a little bit over time. Maybe one or two more questions. What do you think, Henrik? Do you still have time for maybe two more questions?
Sure.
Okay, two more. Does the minimum 3D PSA and further neutral TPSA predict BBB penetration? Coming back to the, I guess, CNS.
I think we touched upon that question. I mean, to have CNS penetration in the first place, you have to have high permeability.
Right.
And that's what this is good at predicting. The efflux ratio is to a large extent SAR, and you may have or not have that in your series. It's a bit like plasma protein binding. Depends a lot on how polarity is distributed and how different pharmacophores are represented. It's not in a way predict. I mean, it has no say on ER.
Right.
That's just SAR.
Right. Yeah, maybe someday somebody will develop something similar. That would be amazing for I guess the CNS drug discovery. But I think there are now ways to predict KPUU computationally, which I think are super fascinating. We'll actually have an article on Drug Hunter coming out pretty soon.
Cool.
Okay, last question. How do you ensure you can trust any biochemical or other in vitro data generated with compounds which have such poor physical properties that the logD cannot be measured?
I mean, first of all, the saving grace is often the presence of protein in these assays. And that also makes a difference in vivo. Of course, the flip side of the coin is that they tend to have high plasma protein binding, and which reduces your free AUC. Of course, you should always mistrust data, any data, unless they make sense. So yeah, you can change the presence of protein, which is often a good practice for these kind of molecules. But once you go in vivo, you're not talking about aqueous solubility, but more biorelevant media solubility and supersaturation. Yeah, I mean, these drugs, they in the end, they work in cells and also in patients. Yeah. But characterizing them in vitro can be very tricky and misleading.
Makes sense. Yeah, and I guess it's often the data that doesn't make sense that gets us thinking. And I think that's how you got to the concept of non-classical Switterions, right? I think you explained that beautifully in your talk at the ACS. Yeah, and with that, we're Yeah, well, more than 5 minutes overtime. I apologize, Henrik. Thank you so much for a phenomenal talk. That was really, really great. I think, you know, people walked away with concepts that they can practically use in their Beyond Rule of 5 campaigns, and maybe even in just normal drug discovery campaigns, frankly, right? So thank you so much. Yeah, thank you so much for you know, giving this excellent presentation and being here with us to answer all the audience questions. Was amazing.
Super. Thank you very much, Dennis and team, for giving me the opportunity to share these concepts here. And I hope they'll benefit patients in the future.
They definitely seem useful to me. So yeah, thank you also to the audience for all your engagement, all your questions. We will see you at the next event. So thank you, everybody. Bye for now.
Bye-bye.